students under supervision of Professor Ryszard Tadeusiewicz and Dr. Patrycja Domalik-Pyzik. And today I would like to present my research on biomaterials enhancing angiogenesis. Angiogenesis is a physiological process of formation of new blood vessels from pre-existing ones, and it involves endothelial cell proliferation, migration, and tube formation. It is very important because in regenerative medicine, insufficient vascularization is one of the biggest limitations, because oxygen can diffuse only to 200 micrometers from the blood. So all the cells that are in a bigger distance than this cannot get oxygen, so they die. So the angiogenesis is essential for tissue growth, wound healing, and regeneration. And here is the scheme of how angiogenesis works. So as we can see, it is a multi-step and highly regulated process. And it all begins with endothelial cell activation, which is triggered by proangiogenic signals such as vascular endothelial growth factor. Those signals are released in response to hypoxia or tissue injury. And the next step is matrix degradation. So the basement membrane is degraded through the activity of matrix metalloproteinases, and it allows our endothelial cells to detach from the vessel. So when they detach, there is the tip cell, the tip endothelial cell, that is forming a vascular sprout. So all the other endothelial cells are proliferating, and they are stalking cells, they are elongating, and because of that, we are formatting the lumen. So it is enabling the development of a functional vessel that is capable of blood transport. And the last step in our angiogenesis is network formation. So it is about the maturation and stabilization of vessel, which involves recruitment of pericytes and smooth muscle cells. Angiogenesis is very important in our health, but it needs to be regulated, because physiologically it is necessary for tissue repair like wound healing or organ regeneration, but it can be also pathological. When there is excessive or uncontrolled angiogenesis, it can support tumor growth. But when there is insufficient angiogenesis, it can lead to chronic wounds or ischemic conditions. So this is very important that the angiogenesis is precisely regulated in medicine, so we can stimulate it in regenerative therapies, and also we can be able to inhibit angiogenesis in diseases like cancer. There are several strategies to support angiogenesis in tissue engineering, and they include the delivery of angiogenic growth factors such as vascular endothelial growth factor or fibroblast growth factor. So we can deliver them either directly or with controlled release systems, and the same applies to bioactive molecules. We can deliver proangiogenic molecules like metal ions or bioactive compounds. And the last strategy, but very important, is the design of proangiogenic scaffold. So our scaffold should provide structural support, and the environment should be easy to proliferate for endothelial cells. So it includes designing a proper porosity of our scaffold, surface chemistry, and hydrophilicity. Biomaterials can mimic the natural extracellular matrix, and it is providing both mechanical support and chemical cues for angiogenesis. What is the most important while designing a biomaterial designed for angiogenesis is that the material should be biocompatible, biodegradable, and be able to deliver bioactive compounds in a controlled manner. So by optimizing all of these properties, biomaterial can create an environment that encourages cell proliferation, migration, and vessel formation. And hydrogels are particularly attractive for all the tissue engineering applications, especially the ones for angiogenic applications, because they have high water content and they resemble native soft tissues. They are permeable to oxygen and nutrients, so nutrients and oxygen can better diffuse, and their mechanical properties can be tuned to influence endothelial cell behavior. Hydrogels can actively participate in angiogenesis by presenting biochemical signal, either through physical encapsulation or affinity-based binding of growth factors. This allows sustained angiogenic stimulation, what is better than like a short-term burst effect when all the bioactive molecules are released in a moment. And why angiogenesis is critical in bone regeneration. So in the picture on the right, we can see a bone that is a highly vascularized tissue. So bone regeneration is highly dependent on vascularization, and blood vessels not only supply nutrients and oxygen, but they also serve as pathways for osteoprogenitor cells and signaling molecules essential for bone repair. It means that impaired angiogenesis leads to impaired bone healing. And as we can see here, those two processes are tightly coupling together because early vascularization in early vascular invasion precedes mineralization. Osteoblasts release angiogenic factors like vascular endothelial growth factor, and endothelial cells can secrete osteogenic signals. So my research motivation was that despite the advances in biomaterials design, the challenges still remain because many scaffolds fail to achieve sufficient vascularization. So my goal was to develop biomaterials that promote angiogenesis effectively by combining both chemical cues and scaffold structure for enhanced angiogenesis. And here is our first project, the first project that I did on my PhD studies. And this was the research conducted in collaboration with Lancaster University, which we can see in the photo on the right. And this study aimed to design protein-based hydrogels for tissue engineering applications where angiogenesis is essential. So as I said, particularly in bone regeneration. This study explored the use of whey protein isolate as a scaffold material and its modification with bioactive additives to improve biological performance. So why did we choose WPI? So WPI, whey protein isolate, was selected because it's highly bioactive, it's biocompatible, biodegradable, and has ability to form stable hydrogel networks. It possesses antimicrobial bioactivity, and it is a byproduct of the dairy industry. So it is easily available and very cheap. We can make sterilizable hydrogels from WPI, which is very important in clinical use, and over 90% of WPI consists of proteins such as beta-lactoglobulin, alpha-lactalbumin, and bovine serum albumin and lactoferrin. We chose as other additives heparin, and heparin is a highly sulfated glycosaminoglican that is widely known for its anticoagulant properties and commonly used in thrombosis treatment. It has the highest negative charge density among biological macromolecules, which enables strong ionic interactions with proteins and growth factors. So at appropriate concentration, heparin has been shown to stimulate angiogenesis, but appropriate concentrations are very important because when the concentration is too high, it can suppress angiogenesis. The other thing we use, the other thing is tinzaparin, and this is like a low molecular weight derivative from heparin, and compared to unfractioned heparin, it exhibits more distinct biological and pharmacological characteristics, including more predictable bioavailability and a longer half-life time. So these properties make tinzaparin an attractive additive for biomaterial-based angiogenic strategies. And here on the right, we have a picture of our samples that we made. So the main component of our hydrogels was 40% WPI, and the hydrogels were modified with heparin or tinzaparin, added at concentrations from 2.5, 5, 7.5 to 10%. And the gelation of those samples was induced in a water bath. We set the water bath at 70 Celsius degrees, and we left our samples for around 5 minutes in it. And yeah, in the right, those are the samples. From the left, it's pure WPI, and then it's 2.5, 5, 7.5, and 10% of additive. Above it is tinzaparin, and our picture shows heparin. Our hydrogels then were sterilized with autoclave, which ensures that they are suitable for in vitro biological evaluation, but it also ensures it is very important that our biomaterial can be sterilized without losing its properties, so it can be translated to clinical applications. And we did multiple compositions to assess the influence of additives content on hydrogel properties. To evaluate the hydrogels, we made a combination of physicochemical and biological analysis. So we checked the structural characterization using FTIR and SEM. We checked mechanical properties and swelling behavior to determine material stability and hydration. And we also did biological assessments that included cytocompatibility, cell attachment, gene expression analysis, and angiogenesis assays that allowed us to know the cellular response and angiogenic potential of our samples. Here are results of our research. So figure 3 shows SEM images of hydrogels. From the left, it's 10% heparin. In the middle, it's 10% tinzaparin. And on the right, it's pure WPI. And as we can see, modified samples didn't have the same morphology compared to the control. They had much higher porosity, which is very good. We wanted to obtain that. And on the right, we have figure 4 that shows FTIR analysis with highlighted characteristic heparin peaks. And we can see that with the amount of additives, with higher amount of additives, those peaks are getting more visible. So both FTIR and SEM confirmed that HP and TP were successfully incorporated into the hydrogel structure. Here we have mechanical properties and swelling. On the left, there is a graph showing average compressive stress at 60% strain. And on the right, there is the average swelling ratio after 5 days of incubation. And it was predicted that additives will lower the hydrogel's mechanical properties as they increased the porosity. Which is also visible in swelling, as increased porosity influenced the swelling of hydrogels, and the maximum swelling was reached at 7.5% additives for both tinzaparin and heparin. Here are some results from our biological assessments. So on the left, there is and cell viability and proliferation of human dental pulp stem cells that were seeded on hydrogels, and absorbance values were taken at days 3 and 5. So what is important from this diagram is that none of our samples had any cytotoxic effect. All of them were cytocompatible. And for heparin, the 2.5% had the highest cytocompatibility, and from the tinzaparin it was 10%. On the right we have the SEM images demonstrating cellular morphology of human dental pulp stem cells on WPI, and all hydrogels with additives improved cell attachment compared to pure WPI. Here we have angiogenic activity, and figure 7 represents cell migration assay. And cell migration assay is when we have a layer of our cells, we make a scratch, then we add our material and we check if our material is enhancing the cell migration towards the wound closure after time, of course. And the results of the scratch assay, and also we made a tube formation assay, confirmed that 10% of heparin and 2.5% of tinzaparin had the highest angiogenic potential, and those results were the same as results from relative gene expression, as the same samples exhibited the highest angiogenic potential in vitro because they had the highest angiogenic markers expressions. So our obtained hydrogels had a positive impact on angiogenic markers, and in previous work they were also reported to enhance osteogenesis. Our next project was conducted in collaboration with University of Crete, which we can see in the photo. And those results focused on mineral-based biomaterials that aim to design calcium carbonate microparticles for bone tissue engineering applications that should target both osteogenic and angiogenic processes. We chose calcium carbonate as a primary component because of its high calcium content, which supports bone formation, because as we know, bone is consisting from mainly calcium. The additives that we used were also heparin and its low molecular weight derivative tinzaparin, because as we knew from our previous research, they can also support both angiogenesis and they have some osteoinductive potential. And we also picked magnesium ions. They were incorporated because from the literature we knew that they can stimulate both vascularization and osteogenic activity. And figure 9 shows SEM images of our microbeads at 3000 times magnification, and upper row is without magnesium addition and lower row is with magnesium addition, and it is clearly visible that magnesium addition was distracting. It didn't made bigger particles, and the particles shape was not like one microparticle, but the whole particle was like a double bell shaped. They are much larger than the microparticles without magnesium. And we chose those methods to check our microparticles. We evaluated crystalline structure, microstructure, and we did some biological assessments towards angiogenesis and osteogenesis. So FTIR and XRD analysis confirmed structural changes that were connected with successful incorporation of additives. Addition of magnesium affected the morphology of calcium carbonate crystals, which we seen on the previous slides. FTIR confirmed incorporation of magnesium ions into calcium carbonate structure, and XRD, which we can see here below, decreased crystallinity with magnesium heparin or tinzaparin, and only so the green dashed lines are for vaterite phase, and red dashed lines are for calcite phase. And calcite and vaterite are polymorphic crystalline forms of calcium carbonate. And calcite is the most stable polymorph, and vaterite is metastable phased, but it exhibits higher specific surface area and higher porosity, so usually is more desirable in tissue engineering. And from our samples, only calcium carbonate and calcium carbonate with heparin contained vaterite. All the other samples contained only calcite. And here are the results of ALP activity. So to make it easier, what is ALP? It is the alkaline phosphate assay that measures the activity of alkaline phosphatase, which is an enzyme that is expressed by osteoblasts during early stages of osteogenic differentiation. So ALP indicates early osteogenic differentiation and functional activity of osteoblast-like cells. So what was for us quite surprising was that under those conditions, magnesium didn't support osteogenic differentiation. But as we can see, all the samples achieved better results than our control. So all of the samples supported osteogenesis, though the samples without magnesium supported it better. Here are some other results of osteogenic assays, and above there is a collagen cumulative production assay, which quantifies the amount of collagen produced by cells over time, and it indicates the extracellular matrix synthesis and osteoblast maturation. And below there is calcium deposition assay that measures the accumulation of calcium salts that are deposited by cells, and it also indicates late stage osteogenic differentiation. So from our results, we can see that here also TP containing samples exhibited the highest collagen and calcium production, which means they supported osteogenic activities the most. And here is also cell migration assay of Wharton jelly's mesenchymal stem cells. So it's another scratch assay results. So we did that to evaluate the angiogenic potential of our samples. And it further confirmed the biological activity of the materials as tinzaparin functionalized samples showed the fastest scratch closure, so suggesting enhanced proangiogenic behavior. So overall, tinzaparin functionalized calcium carbonate materials showed the most promising biological performance, and they both combined enhanced angiogenic and osteogenic indicators. So, yeah. Now our next project was prepared here on AGH, and it was about alginate microbeads enriched with calcium carbonate, tinzaparin, and copper for wound healing applications. So alginate is a widely used biomaterial due to its biocompatibility, high water absorption capacity, and ability to maintain a moist environment. And calcium carbonate, as we know from before, provides a source of calcium, supporting tissue regeneration and cell proliferation. Copper ions are well-known proangiogenic and antimicrobial agents, which can enhance vascularization and prevent infection. And as we know from the two previous researches, tinzaparin can also stimulate angiogenesis. So incorporating these microparticles into alginate hydrogels combine the structural and moisture retaining advantages of the hydrogel with the bioactive effects of calcium carbonate and copper and tinzaparin, potentially accelerating tissue repair and improving healing outcomes. So we made 3 formulations, and all of them were cross-linked with calcium ions. It was pure alginate with calcium carbonate, and alginate with calcium carbonate and tinzaparin. But we also cross-linked pure alginate with copper ions to test what will it change. And the test we did was morphology, water uptake capacity, shrinkage upon drying, and FTIR. And here are our results. So microscopic analysis showed uniform microbead morphology. And as we can see, so from the left, it's pure alginate with calcium, then it's alginate cross-linked with copper, alginate with calcium carbonate, and alginate with calcium carbonate and tinzaparin. And as we can see, the microbeads that were cross-linked with copper had the roughest surface, and also they had the highest water uptake and the biggest diameter, both before drying and after drying. So the conclusions that we took from those results are that the cross-linking ion and additives strongly affect bead properties. As we can see, the alginate that was cross-linked with calcium was the most stable, while alginate that was cross-linked with copper was the most hydrated and shrinkable formulation. So those results are promising for wound healing, but of course, further studies are required. And the most recent research was conducted thanks to the Dean's grant from our faculty, and I could make an internship in Poznań University of Medical Sciences. So I went there for an internship, and with the team from there, we were choosing the best biomaterial that could support angiogenesis and wound healing, and we chose Centella asiatica, which we can see in the picture on the right. This is a medicinal plant that is known for wound healing properties. It stimulates the collagen synthesis, angiogenesis, and ACM matrix modeling. Incorporating Centella asiatica into alginate hydrogels combines both the structural and moisture-retaining advantages of the hydrogel and the bioactive wound healing effects of the plant. And it could potentially result in a wound dressing that would protect the wound and actively promote tissue regeneration. And what we did in Poznań was comparison of 3 different extractions methods, and we did maceration. So for maceration, this is a classical method for gentle release of bioactive compounds, and we left our extract for 24 hours at room temperature in darkness. The other method was heat-assisted extraction in a water bath, so we left our extract for 1 hour at near boiling temperature, and this method uses high temperature to enhance solubilization and release of compounds. And the third method was ultrasound-assisted extraction, where we left our extract for 1 hour in ultrasound bath, where ultrasound waves improve the solvent penetration and accelerate the release of bioactive compounds. And the biological activity assays that we chose to do was DPPH assay that evaluates antioxidant activity, so the ability to neutralize free radicals. We checked hyaluronidase inhibition, so it tests the impact on hyaluronic acid degradation. We did iron halation, and it measures ability to bind metal ions, so it protects against oxidative stress. And we also did Folin assay. It determines total phenolic content related to antioxidant potential. So our results are, I'm still analyzing the results, so I cannot show them yet. But from what I've seen from the results, the best method of preparing the extract was ultrasound-assisted extraction, as it had the highest biological activity in all of these 4 assays. And what will we do now after analyzing all those results? So what are we doing now, basically, is taking those extracts and putting them into alginate hydrogels, from which we would like to make patches. So it will be like a wound patch that we will put on a wound as a wound dressing. And while alginate will degrade, it will release Centella asiatica extract that should enhance the wound healing and the angiogenesis processes. What are the future plans for my PhD thesis is that we are planning to develop a dual-phase 3D printed scaffold with a microchannel architecture that is designed to support angiogenesis. So basically this concept is based on combining 2 biomaterials with different degradation rates. So the faster degrading phase is intended to create a microchannel networks upon degradation, and those microchannels should serve as pathways for newly forming vessels. And while degrading, this material should release some pro-angiogenic molecules. And the second, slower degrading phase, is designed to provide structural support and sustained release of bioactive components that may promote the vessel stabilization and maturation. So this dual phase strategy aims to mimic key aspects of natural angiogenic process. And of course, this work will include evaluation of scaffold biocompatibility, degradation kinetics of both material phases, release profiles of bioactive molecules, and of course, standard in vitro assays, like we seen before, to evaluate the cytocompatibility or angiogenic potential. I would like to thank all my collaborators from Lancaster University's Professor Timothy Douglas, Daniel Baines, and Professor Alan Smith. From University of Crete, it's Professor Maria Hatzinikolaidou, Barbara Platania, and Nicoleta Tavernaraki. And from Poznań University of Medical Sciences, it's Professor Judyta Cierecka-Piątek, Dr. Magdalena Paczkowska-Walendowska, and Dr. Elżbieta Studzińska-Sroka. I would also like to give my thanks for all the fundings that made it possible to do all this experiment and researches. So it is EDUP Research University Excellence Initiative and our faculty of Electrical Engineering, Automatics, Computer Science, and Biomedical Engineering for the Dean's Grant, and also the Faculty of Material Science and Ceramics at AGH, where I do mostly all of my experiments. And that's all. Thank you for your attention.
Thank you very much for the interesting talk. And at the end, I would like to thank you for the activity. And I think not only in fundraising, but I also noticed at least 4 papers prepared with the help of host universities like Lancaster, Crete, and Poznań University. That's an image of an excellent PhD student, I hope. And if you don't mind, I will give your example as a pattern to follow for all PhD students.
Thank you. I think that's really, really nice and encouraging.
Yes, it was. Your example is encouraging. That should be disseminated as a pattern to follow. So now the talk is open for discussion. Please ask questions. My first question is, once you told about the microstructure evaluation, and you demonstrated several pictures that they look very nicely, and the granularity was seen to be different. Mm-hmm. My question is whether there is a quantitative method like a visual system or some statistics to evaluate such structures automatically.
So we didn't do it with WPI hydrogels, as we just wanted to show that the additives were incorporated. And all it took was just to take a SEM photo, because we could see that already there was a different microstructure. We didn't count any porosity or anything like that. But of course, it is possible. But in this experiment, we didn't use it.
Okay, thank you. Other questions?
Question might be proposition to the last subject. You showed us the scaffoldings, printed 3D scaffoldings. My proposition consists of natural scaffoldings, because 3D scaffoldings characterized by relatively claim dimensions, but in contrary, the natural scaffoldings, I mean, prepared from plants, from leaves or roots, emphasize more natural structure. And such structures are relatively often used as such scaffolding. So, what do you think about involving such type of scaffolding in your experiments?
I'm sorry, but I didn't hear it very well.
So, again,
Maybe you can come closer. Yeah, here is the microphone.
So I don't have a question, another proposition, because you have presented the application of 3D scaffoldings. And I would like to ask you about the possibility of taking into consideration in your investigation also the a little bit different scaffoldings which are obtained by preparation the natural plants, mostly. Leaves or roots, so it is a possibility to obtain more natural, in much more lower scales scaffoldings. So, what do you think about the possibility of about extending your results into use such type of scaffoldings?
So yes, I would like to work we work mostly with natural polymers, so like WP.
I understand, but this is a scaffolding. Usually it is, it's difficult to say made of cellulose because it is natural origin. But it is very fine structure originated from natural organisms.
Mm-hmm. Yes. So
you easily can adjust the scale of this scaffolding to your expectation because you could use different plants.
Yes. So
I don't know how it is in English, but for example, copper polish, it's a special plant used in the kitchen, and very often it is it is swollen to obtain cellular structure.
So yes, so I'm not sure if I understand correctly, but
it is not a question. I'm not a specialist in microbiology, but I have seen very often such scaffoldings which are very easy adjustable to certain dimensions expected by application of the certain chemicals, particles.
Okay.
It's not a question.
Okay. No. I understand. So that you mean that you would use those materials also as scaffolds on themselves. Yes, that's themselves. So decellularize the plant and use. Yeah. Yes, I know about this way, and yes, we could use it. But what I have planned in my in my plans for my PhD was the 3D printing scaffolds. So that this is why I will now concentrate on that way.
It is very simple proposition because it is relatively easy to decellularizate, but it is a wrong name because it is not decellularization. Of course, it is a very often used name, but the results of this process is cellulosis in the structure. So the process calls decellularization, but the results, it is a structure made of cellulosis. But never mind, it's only a proposition.
Thank you very much. I will consider it for sure with my Dr. Domalik.